PowerPoints because, in essence, what we're doing today is reviewing for the exam, right? So that's a good thing. So let's talk some details about mechanics of the test and the quiz and stuff like that. Answer questions about that, and then we'll figure out how we would like to do some review today. To be sure, we can go through, if you have pressing questions, you're like, oh yeah, I looked at things, I was reviewing things, I really don't get this, can we go through this topic? We can hit those first. We can just kind of scroll through the PowerPoint slides quickly and remind ourselves about kind of bigger topics as we were going through them as well. I do have some kind of example multiple choice questions that we can do together and have that kind of jog our memory about particular topics that may be important or particular questions that we may have. Anywhere we want to go with this, we can do that, right? This is our time. That's the whole reason why we do the exam online, to give us a little bit more time to review, okay? And the reason that we're doing that is, of course, our lecture exam is a couple days away. It's due at 11:59 on Saturday, which, in terms of the schedule, 2 days before we have a lecture exam, we always have our lecture quiz due. So the lecture quiz, in terms of the format, will be all multiple choice, 20 multiple choice questions that cover our different chapters that are going to be covered on the exam. So we're covering the same content on the quiz that we are for the exam. Now for the quiz, that one is only multiple choice. So we try to make it easier in a few ways. It's only multiple choice. It is technically open book, but you will be using Respondent's Lockdown browser with the quiz and the exam. The exam is closed book, closed note, closed other resources, etc., right? Assessing what you've learned, coming only from your own mind. But the quiz is just kind of a checkpoint for you to see how you're doing with all of the content that we have covered and we're expected to be assessed on for the exam. So with that one, we're using Respondent's Lockdown browser just to make sure everything with the Lockdown browser is working fine on your device, right? So that way, when you get to the exam 2 days later, it's not a worry like, oh, how do I do this? Or I don't know, my camera isn't working or whatever. We just checked it when we did the quiz and it should be good to go. So that being said, do practice going through the initial setup steps for Respondent's Lockdown browser. So it has you take a picture, it has you do the ID, it has you do most notably your environment check. So you want to take that camera and you want to show your area, pan slowly around, and then make sure that you take that camera 90 degrees and show your desktop area, right? You've got to show your workspace wherever you are. Now, it's totally fine if that's the kitchen table or the desk in your room or the desk in the library, if you decide to do it here or something, or the couch or wherever you want to do it. Just make sure that there's nothing around, right? So your phone shouldn't be there, no notes should be there, or anything like that, right? It's just you and the test. So once you do that, then put the camera back up, make sure it's adjusted so that your face is visible in it again, not like this or anything like that, because then it's going to be sending you notices that it can't see your face during the exam. And then once you see you're back in the camera again, good to go. Go ahead and start the test and just forget about all that, right? So do that for the quiz as well as for our exams, because I'll make sure to go through the videos on the quiz as well, even though it's open book. That way I can make sure that everybody is doing that environment check properly before we get to the exam itself, okay? With the quiz, like we said, it's open book, open notes, whatever. Of course, you're going to be doing it on an electronic device. So if your notes or anything that you want to consult is also on an electronic device, for the quiz, it's open book. So it's okay for you to have your phone there or your printed notes there or a second device there, right? So if you're using your iPad for the quiz, you can have a laptop over to the right so you can double check the, whatever, the PowerPoint's online or something like that. But because you're using Respondent's Lockdown browser to take the quiz, you're not going to be able to access anything else on that electronic device during the quiz. So just plan ahead of what other type of information you want to have to consult with during that quiz, okay? And as I was mentioning before, what I would suggest you do, treat it like it's closed book, right? That's due tonight. you're kind of working your way, so you should be. We finished up our hemodynamics and blood vessel chapter last week, so we should have been doing some preparing already for all 3 chapters that are on the exam. But do your best on the quiz to at least do your first attempt at all of those questions just out of your head, right? What's the answer for this? Put in answers for all of them. And then with any time that's remaining, because it is time, with any time that's remaining, it's okay to go and check and oh, I'm not sure about that one. I'm going to go check my notes and double check that one before I submit it, okay? But if you do that, where you're doing your first attempt at it, this is my best guess, when you're double checking those, if you have to change them before you submit it, that's okay. But just note that mentally. It's oh, those questions had to do with blood pressure and hemodynamics. I seem to miss a number of those. I had to change a number of those. So in my next couple days leading up to the exam, that can help to guide me and say, oh, okay, well, maybe that's an area where I need to study more. The blood questions I was okay with, the heart questions for the most part, the anatomy stuff I was good with. Use that to guide your next 2 days in terms of how you want to continue preparing, okay? Make sure that you go over your vocabulary list as well as your exam objectives also, right? So those can be found. Here that says a review. Here's our exam 1 material, right? Blood, heart, blood vessels. We can see some of our study materials down here, the objective sheet, the vocabulary list. Those you want to be consulting. And I do have those up and downloaded as well if we wanted to take a look at those, if you had any questions about those too. Remember that, what you're seeing here are the major topics for each of our different chapters. So there's heart and blood vessels and hemodynamics and stuff like that. So when you're looking through those, you should at least, in your head or whatever, or talk it out with a friend or write down your answers to those because those are the major things that we covered. But the ones that are in bold are your possible short answer questions as well, right? So for those, not only do you want to be able to recognize those if you saw it in a multiple choice question, you could, do matching in essence. But for a short answer question where it just asks you about it, you'd be able to put out a complete answer, write out a complete answer describing that particular topic, that physiological process, comparing and contrasting things, etc. Okay? So you want to do some kind of extra planning with those. Most of your content that you're going to need to know for the exam, you'll find in the PowerPoint handouts. Of course, as we go through it here in class, we're always talking, and there's a little bit more detail, right, that we add in addition to those outlined points that we see on the PowerPoints. But most of what you need to know, you can find in those PowerPoints in combination with our Panopto recordings or whatever, right, your own notes that you've made along the way. So, the core of the content you'll need to know for the exam is right there. Now, as we mentioned, you should have some sort of a textbook as well that you've been kind of doing your reading on, maybe afterwards and looking at the tables and figures and stuff like that. So that helps to fill in any extra gaps or, give a different explanation of a particular topic that maybe is a little different than what we talked about here in lecture. But the core of the information that you have is what we have together when we're here in class, right, in the lectures, etc. So there's not going to be too much outside of that. There might be finer details or something, but really you should be able to find answers to all these within what we've discussed. Okay? Okay. So, questions. Questions about any aspect.
If you could go over the ECG
Yeah.
again.
Yeah, for sure. So the good thing about that is that's one of the things, of course, that we do in lab this time as well. So we're doing physiological tests in lab. So we are going to be talking about ECG and blood pressure and things. Those are things that we have overlap with in lecture too. So it's nice that we have that kind of reinforcement this week too. But let's take a look. So that's going to be in our heart chapter. And we do know that that's one of our bolded exam objectives, right? We're looking at the ECGs. Now, one thing that I want to remind you is that when we look at those electrical outputs, we also had these types of graphs as well that are looking at electrical outputs, right? We see the same labels on the graph. We see time on the x-axis. We see millivolts on the y-axis, right? But remember, when we were talking about these graphs, we were saying these are the changes in membrane potential of our two different cardiac muscle cells, our contractile cells and our autorhythmic cells. And we're talking about depolarization and repolarization and what ions cause each part of the graph that we're seeing there. And that's really looking at what's happening on individual cells, what's happening in terms of changes in electrochemical activity across their membrane when they're becoming excited and stuff like that, okay? So when we're talking about the ECG, that's a little bit different. With the ECG, what we're talking about is the heart overall. So we're not talking about individual cells. We're looking at an output that shows us these little spikes, if you will, that occur in electrical activity during a cardiac cycle when we're talking about the atria filling and then contracting and the ventricles filling and then contracting. Those cycles that are going through the atria ventricles in a cardiac cycle, we're seeing that on an ECG in terms of spikes of cellular activity. So we've got 3 repeating waves that we need to know. First little one is the P wave, big middle one, QRS wave complex. Last one is called the T wave, right? So for those, as this graph indicates, we need to know what is occurring in the heart during each of those active waves. So during the P wave, we have atrial depolarization. So atrial depolarization, you can see it at the bottom there, where it's taking part in that curve. We can see that the stimulus to contract is spreading to those cells of the atria during the P wave, okay? So the atrial cells are getting that signal telling them to contract during atrial depolarization. Now, after that wave, in that little flat interval, then the atria are going to contract, right? Right now, during the wave, the signal is spreading. They're getting their signal. And then afterwards, they're going to respond, and they're going to contract. They're going to shorten, and they're going to squeeze those atrial chambers, okay? Then we get to the QRS wave complex. That's when now the signal is going to go down to the ventricles, and they're going to get their stimulation to contract. So we see it spreading throughout the ventricles there, and so during the QRS wave, we have ventricular now depolarization as the signal's arriving there, but we also have atrial repolarization, where the atria are going to go into their relaxation and filling period. Because remember, between the atria and the ventricles, they're doing opposite things at different times, right? When the atria are contracting, the ventricles are relaxing, and now when the ventricles are contracting, the atria are relaxing and filling again, okay? So during the QRS wave, we have atrial repolarization occurring, and then we have ventricular depolarization. They're getting their signal. And then finally, when we get to our T wave, that's when we're going to have our ventricular repolarization as well. So for the QRS wave and the P wave and the T wave, all these aspects of the ECG, you need to be able to say that we have those are the 3 repeating waves. We need to be able to say what's going on electrically during each, right? The atrial repolarization or depolarization, etc., that type of thing. And then, basically, right after that, the mechanical response is, okay, are the atria contracting? Are the atria relaxing and filling, etc.? That happens right after that signal. So after depolarization is when they contract, and after repolarization is when they relax and fill again. Okay? Now, normally, if we were doing like a paper test, you would probably draw it out. You would draw an ECG output. You'd label the axes, and then you say P wave and Q wave, and then describe it. When we're doing it electronically, you're not going to be able to necessarily draw, so it's just going to be a written description of what are the 3 waves, what's going on during each of the waves, and then what's the response afterwards. Right? You're just kind of writing that out. If you get that short answer question. How's that?
Okay, thank you.
Better? Okay. Other things. Topics. Yeah?
How do you find the duration?
The duration. So, that's a good question. So that is something that we will be doing in lab more than lecture. So you won't be asked to do that on the lecture exam. You will have a possible question like that on the lab practical. So with that, and we'll talk about that a bit more in lab, basically, duration is the width of them, how long they take to occur. And then amplitude is basically the height of it, right? How high it is. And that's kind of a measure of like how much of a response are we getting, right? So in terms of duration, when we have it on a typical EKG, you'll have it coming out on graph paper, and you'll see these little units, these little 1 millimeter boxes. And so you count over the number of boxes, and then you have this conversion factor that we discussed in labs, in the lab manual as well. But basically, every 1 millimeter in the horizontal is equal to 0.04 seconds. And every 1 millimeter box in the vertical is equal to 0.1 millivolts instead. So if you have a graph and you see the little 3 little waves, you're just counting boxes, number of boxes, 1, 2, 3, 4, 5, from the highest point to the lowest point per amplitude, or from where it kind of starts and then stops for duration, and then you just convert it. Like if you have 5 boxes high, then basically it's 5 times 0.1 millivolts, so it's 0.5 millivolts in terms of amplitude or duration. I'm sorry, amplitude or height. Yeah. So, but that's more of a lab type of question. Yep. Other things.
Can we go over the pathway of the blood, how it goes from the heart to the lungs? And
Yeah, for sure. That's a good question. So when we have a system where we have a lot of movement through different organs, one of the ways that we can review kind of the different parts and how we're going to move through that organ or move through that organ system is we can do like a tracing. That's what I call it, a tracing, or basically following the pathway of that substance, in this case blood, through the system. So we'll see this with cardiovascular system. We'll see it when we get the digestive system. We'll trace food through all the different digestive organs, etc. We'll do it with urinary system as the urine is created inside the kidney and how it passes through different parts of the kidney and out through the ureter and stuff like that. Bless you. We'll do it for reproductive system as well. Like when gametes are produced, where are they produced? What are they transferred next? Where do glands come in and stuff like that? Okay. So it's really just a listing of anatomical structures that whatever we're talking about, in this case blood, a blood cell, is passing as it goes from point A to point B. So if you are answering a question like this, you can actually answer it similar to how I have it here. You don't have to like write complete sentences or anything like that. You can just basically say this structure to that structure, to that, to that, to that, to that. Okay? So that's fine. You're just listing those structures in order. So for instance, if we're talking about the flow of blood through the heart, now this includes both the right side and the left side of the heart. Now to be sure, when we're doing a tracing, we're thinking about, okay, it moves from the right atrium to the right ventricle, and the right ventricle, right? We can only do one thing at a time. But don't forget, both atria are contracting at the same time, sending it to both ventricles. Both ventricles contract at the same time, ejecting it out of the heart. So there's blood leaving the aorta, and there's blood leaving the pulmonary trunk at the same time. So when we're doing this kind of tracing on the right side and the left side of the heart, don't think that it like marches through the right side, then it marches through the left side. It's definitely going both sides at the same time. Okay? Okay, so if we're thinking about a blood cell that's coming back from the body, deoxygenated blood, and it's going to go through the right side in order to go to the lungs to pick up oxygen again, we're going to arrive at that right atrium. Think about the models that we have in lab, right? We have those two vena cavae, superior and inferior vena cava, that are going to drop blood back into the right atrium. This is deoxygenated blood, right? Right side of the heart, blue blood. So we have those two major veins dropping into the right atrium. From the right atrium, it's going to pass through that right atrioventricular valve down to the right ventricle. Right ventricle is going to contract, and it's going to send it out. It's going to the lungs, so it's going to go to that, past that pulmonary valve into the pulmonary trunk. And the pulmonary trunk is going to split, right, into a left pulmonary artery and a right pulmonary artery to head to the lungs. So it gets to the lungs, and now we're going to do our oxygen and carbon dioxide exchange, and then it's going to head back. So that's kind of the right side deoxygenated blood tracing. Talking about how it gets to the atrium, goes down the ventricle, and then exits out the pulmonary side to go to the lungs. Okay? You want to list the valves along the way too, because that helps to make sure that the blood is going one way and it doesn't go back the other way. Yeah?
Does it matter if this was hypothetically a short answer question, does it matter if we say tricuspid valve or tricuspid valve, or can we just say atrioventricular valve for either side?
That's a good question. When we have, so we're finding out that there are a number of structures around the body where they may have more than one name, and those names are both correct. So for instance, on the right side, we have the right atrioventricular valve. It's also known as the tricuspid valve, okay? Both of those names are correct, so you can use whichever one you like. That's totally fine. If you use atrioventricular valve, you have to put right and left because the name is the same, right? Correct? I don't want to say right? So if you use atrioventricular valve, you have to say right or left. If you use tricuspid and bicuspid, those are unique names. You can just say tricuspid and bicuspid valve. Bicuspid is also known as the mitral valve, so you can use that one as well. Okay, it's totally fine to use either name. So any place you have an anatomical term that you see like in lecture or from a textbook or something like that, that's okay to use that term, right? That's an official term, even though there may be an alternate or something like that. Yep. So that's fine. So left side, now we have pulmonary veins, right? Because veins are going back to the heart. Pulmonary veins are going to return that oxygenated blood to the left atrium. Going to go through the left atrioventricular valve, bicuspid valve or mitral valve, down to left ventricle. Left ventricle contracts, sends it past the aortic valve and out the aorta to go to the body. So that's your left side tracing of oxygenated blood from the lungs to the body. So you got to recognize that as well, right? So recognize if it's asking you about deoxygenated blood, we're talking about the right side. And if it's asking about from the body to the lungs, that's also the right side, right? If we're talking about the left side, that's going to be oxygenated blood, and that's going to be from the lungs, right? Goes into that left atrium, and then it goes out to the body. So from the lungs to the body is going to be left side. So you just got to be thinking about those different circulatory pathways and what that means in terms of, are we talking about the left side circulation or the right side circulation? Because we'll usually just ask you one side rather than all of that in one question, usually. Okay? Other questions? Now you got to think back all the way to blood, right? So blood chapter that we did when we didn't know each other, that was all included in this exam as well. So think back to the blood, all of the different components of the plasma. What are the different things we can find in the plasma and why are they there? What are their function? Different types of ions, water. That's a big one. That's most of the plasma is made up of water. It's a solute or solvent and stuff like that. So you can say water is an important part of the plasma. We have 3 different plasma proteins that are the main ones, right? Our immunoglobulins, like our antibodies. We have our fibrinogen, which is part of that clotting cascade, which is another big topic that we covered in the blood chapter. And then we have albumin, which came into play in terms of osmotic balance, right? It helps to draw water from the tissues back into the capillaries. So you have to be able to think about the plasma component as well as the cellular elements. Don't forget, cellular elements, you basically have like 7 different choices, 7 different cell types that we find in the blood. 5 different types of white blood cells. Each one has a different, slightly different function, plus red blood cells, plus platelets, right? So 7 different things, 7 different cellular elements that you need to know about. Be able to describe, okay, why are they there? What do they look like? Where do they come from, right? What's kind of their life cycle? Where do they come from? They all come from the red bone marrow, so that's nice. They all have that same answer. What are they there for? They all have different functions, platelet plugs or carrying oxygen or bodily defense, etc. And then we also talked about how does the red bone marrow get signaled so that it knows when it's time to produce more red blood cells or produce more platelets or whatever, right? There are specific chemicals that are released that will tell the red bone marrow when it's time to produce a specific red blood cell or white blood cell or something like that. So we had erythropoietin, right? Erythro means red for the red blood cells. We had basically what are called cytokines or interleukins, which is a group of chemicals for our white blood cells. And then we had thrombopoietin as the chemical that tells the red bone marrow to produce more thrombocytes, right? That's their official name, is thrombocytes. So thrombopoietin for thrombocytes, okay? That also reminds me when we were talking about blood cells, we have that big old family tree as well. So they come from the red bone marrow. Starts with a precursor cell, a stem cell that's called the hemocytoblast, right? Hemo with the blood, cytocell, blast is one of those early precursor cells that's really active. So it starts with the hemocytoblast, that gives rise to any of our different cellular elements. From there, that hemocytoblast can divide into either a myeloid stem cell or a lymphoid stem cell. The lymphoid stem cell can only become lymphocytes. The myeloid stem cells can become anything else. All of our other white blood cell types, our red blood cells, and our platelets all come from that myeloid stem cell line. And then for the most part, with the platelets, we said that the last precursor cell before they actually become platelets is called a megakaryoblast, right? This really huge cell inside the bone marrow that gets shredded to produce those thrombocytes that are floating around in the blood as they exit. And then for the red blood cells, we said there's one precursor cell stage we need to know. It's called the reticulocyte. The reticulocyte is the last kind of immature stage where it's kind of giving up its nucleus and starting to get that very unique kind of biconcave disc shape, as opposed to just kind of a generic cell. Okay? So do look at that family tree slide. That shows you the stages that you do need to know in terms of how those different cells are produced and what were the precursor cells leading up to them. Okay? Other things. Yeah, for sure. That's a good one. So there's two aspects of that. So when we were talking about the blood vessel chapter, we did talk about hemodynamics a bit. That was more blood pressure, blood flow, and stuff like that. But one aspect of that, as well in terms of kind of hemodynamics, has to do with hemostasis. So hemostasis is a particular type of homeostasis where we're trying to maintain our blood in the blood vessels and not let it leak out, right? Because then we're going to have problems with blood volume and blood pressure, etc. So there's two aspects of that. So as we get down to the end of that chapter, there's some of that family tree that I was talking about in terms of those cells. This one has all of the different relatives we see right there. But when there is an issue in terms of hemostasis and a broken blood vessel, we have kind of two things that we can describe. One is basically the three phases of hemostasis. First one is vascular spasm, second one is platelet plug formation, third one is coagulation. So for larger blood vessels, these are the three things that happens in that blood vessel when it's broken. Okay? Vascular spasm, so it's got smooth muscle in the wall. It's got smooth muscle in which tunic? It has three tunics in a wall of a large blood vessel. The media, the middle one, right? So we have tunica intima, tunica media, tunica externa. So you need to know all three layers of a typical large blood vessel and what makes up each of those layers. That's something we do in lab and lecture, okay? It's also part of when you're comparing your different blood vessels, how you can compare and contrast them. So this is a small aside. When we talk about blood vessels, right, we have a lot of different blood vessel types. We have arteries, arterioles, capillaries, venules, and veins. So there's 5. But don't forget there's subcategories. In arteries, we have elastic arteries and muscular arteries. In capillaries, we have 3 different types of capillaries. Continuous capillaries, or sinusoids, as well as fenestrated capillaries. So when you're doing comparisons, there's like 10 different specific types of blood vessels. If you were asked to compare and contrast them, each one you could say, okay, this is what their structure is like, and this is how it relates to their particular function, right? Sinusoids, those capillaries have huge holes in their epithelial wall as well as their basement membrane, which makes it easier for cells and large molecules to get across the wall, right? So you're explaining why their structure looks that way and how it relates to their function. But you've got like 10 different choices. On a short answer question, I'm oftentimes only going to ask you a subsection of that, right? I'm not going to ask you every single thing. So for blood vessels, I may ask you to compare and contrast 5 different blood vessels in their structure and their function. You can choose any you want. You can do those 3 capillaries and the 2 types of arteries. Or you can choose just the basic types, like we said, arteries, arterioles, capillaries, venules, veins. Whatever combination you want, you can do. And that's also one of the reasons I put the short answer questions in there, because it gives you flexibility about what you tell me about what you've learned, as opposed to just straight up multiple choice question, get the right answer or you get no points, right? Yeah.
How many short answer questions are on the exam?
So typically we'll have, I think the layout is this. Typically, we'll have about 20 multiple choice questions. Each one worth 2 points, right? So 20 times 2, no, 40. You have to have 40, I think. I think we have 40 multiple choice questions. So most of the exam is going to be multiple choice questions. So about 80% multiple choice. We usually have 5 vocabulary questions. So of that list of vocabulary terms you have, you're going to see 5 randomly chosen and given to you, where you'll provide a definition, right? There's the word, give me a definition for it. And again, usually about 1 sentence is fine, but just make sure it's specific enough that I'm not going to get it confused with other terms, right? So if we're asking you about a specific white blood cell, I don't think that's on the list, but if I ask you about an eosinophil, don't just tell me it's a white blood cell that defends the body, right? That would define any of our 5 different types of white blood cells. It's got to be specific enough that it defines that particular term. Okay, so 5 points of vocabulary, and then usually we have, I think, 15 points of short answer. So that usually means that there are 3 short answer, bless you, 3 short answer questions, each one worth 5 points, okay? And we know that in the exam objective sheet, those bold ones, that's where they're going to be coming from. So you're probably going to see a combination, again, it's random as to which one you get, a combination of 3 of those questions having to do with those bolded exam objectives, like ECG, like we were talking about, or different blood vessel types or something like that. Okay, so about 15 points of short answer. And that reminds me as well, before we finish up this, right, these 3 different steps, is that for the grading, what's going to happen is you guys are going to submit it. For the multiple choice questions, Canvas is going to be able to automatically grade it. But for the vocabulary and for the short answer, right, those are kind of free-form answers, so it's not going to grade those. It's going to wait for me to grade those. So it's going to give you a grade immediately, but realize that's just of the multiple choice section. That's not your total grade. So don't panic, right? So if you got whatever, a 70 or something like that, that's not a 70 out of 100, that's a 70 out of those 80, and now we have an additional 20 points possible that I'm going to be grading basically next week, okay? So that's only a partial score when you immediately see that score, okay? And then I'll be doing grading next week to finish up those other sections. So don't panic, yeah?
Can you explain like a protracted cardiac action potential?
Yes. Yeah, so we definitely can do that. Let's finish up this one, then we'll move on to that. So vascular spasm, we said, of the 3 steps for hemostasis, vascular spasm is where they're constricting and dilating, if they have that tunica media. Platelet plug formation, platelets, their whole job is to be circulating around, looking for when there's a broken blood vessel. And the way that they know that there's a broken blood vessel is that they're actually seeing exposed collagen fibers sticking out, kind of like rebar sticking out of a building or something like that, right? You're not supposed to see that. It's supposed to be deep in the wall, but we are seeing collagen fibers sticking out. So if they see that, they're going to stick to the collagen fibers and they're going to stick to each other and form that platelet plug, okay? So the first 2 steps in this hemostasis are pretty, self-explanatory or basically simple to describe. A couple sentences you could describe those. The coagulation of the blood clotting, that 3rd phase is a bit more complex. In that one, what we would be talking about is basically what we see over on the right-hand side here, right? So we see that there are 2 clotting pathways. And so this is another short answer question where it talks about blood clotting or coagulation and the difference between the extrinsic pathway and the intrinsic pathway. So it's kind of 2 separate things, and there's some overlap between them, and there's some differences between them. So we have an extrinsic and intrinsic pathway. So the first thing is, where do they occur? So the extrinsic is going to be outside of the blood vessel, exterior to it, right? Intrinsic is going to be within the blood vessel, in the wall. So if we have a broken, dam or levee or a pipe or something like that, we've got to repair the pipe, but we also want to make sure that we're kind of sandbagging outside of it so that whatever's leaking out isn't leaking into the floor of the house or something like that, right? So extrinsic is outside, intrinsic is inside. Extrinsic, the thing that starts it off, is basically things from the tissues outside of the blood vessel, like tissue factors, that will then stimulate the whole clotting cascade to occur. Intrinsic is when we start with the exposed collagen fibers, like we talked about in terms of that platelet plug inside of the blood vessel. So what gets them started is a little bit different. Tissue factor versus collagen within the broken wall of the blood vessel. Okay? If we continue on, there is this whole complex clotting cascade where there are these clotting factors, these proteins that are floating around in your blood, and one activates the next one, activates the next one. There's like 13 of these, like dominoes. One goes this one, that one, that one, that one. We don't have to know all that, okay? But what we do need to know is that the intrinsic pathway basically uses the entire domino cascade of all of these different clotting factors. So it uses all the clotting factors, and the extrinsic only uses some of the clotting factors. So it's much faster, the extrinsic is much faster compared to the intrinsic. And that makes sense because if we're flowing into other tissues, now we're starting to damage other tissues as the blood's leaking out and stuff like that, and we want to kind of rein it in. We want to stop that flow, so we want to make sure that we can do that very quickly, like I say, kind of like throwing down sandbags. So they both use a clotting cascade, and certain clotting factors, we can see, oh, okay, there's clotting factor X there, that's 10, etc. But the extrinsic is faster because it uses some of the clotting factors, whereas intrinsic is a little bit slower because it uses all of the different clotting factors. But ultimately, what the result is we get this enzyme here activated, prothrombinase, see it's an enzyme, ASE, that gets activated, which then activates thrombin, and thrombin, as it gets converted, activates fibrinogen. So fibrinogen gets converted into fibrin, which then fibrin threads are layering onto that platelet plug, which forms a solid clot, okay? So we see at the beginning some differences between these. Where do they occur? How fast do they occur? Do they use all of the different factors or not? But then at the end, we see that the result is the same, where we have the activation of fibrin to form those fibrin threads to form a nice solid clot so that we can stop that loss of blood into the tissues or out of the blood vessel, etc. Okay? So that part, when we're talking about the clotting cascade, that's a little bit more detailed than just, oh, vascular spasm and platelet plug, jamming some platelets in there as a temporary fix, right? So you'll want to review a bit of that in terms of those two clotting cascade pathways, extrinsic and intrinsic. Okay. So then we were also asking about, in the heart, we were talking about those action potentials. And so that's a bigger topic as well. We discovered that there were two different types of muscle cells, right? So we have our cardiac muscles, which are the contractile cells, and then we have the autorhythmic cells. Two different types of cardiac muscle cells. So with those, in a compare and contrast, we want to be able to name them. What are the two types of cardiac muscle cells? Those are the two types. Talk about how frequently do they occur? Well, there's a small percentage, right? Less than 1% of the autorhythmic cells, the manager cells, don't need a lot of managers, need a lot of workers, right? The contractile cells are the workers. So over 99% of our cardiac muscle cells are contractile cells. So we can compare them in terms of how many do we have around? Are there a lot, a little, etc. Then we can also compare them in terms of where they're found. So the autorhythmic cells are going to be found within the conduction system of the heart, like in the SA node and the AV node and the bundle branches, Purkinje fibers, stuff like that. Meanwhile, the contractile cells are going to be found everywhere else within that myocardium. So when we see this diagram, all that you see in yellow there is going to be made up of autorhythmic cells, only in those places. Whereas all of the thick muscular walls of the heart is where we're going to find our contractile cells instead. Okay? So some comparisons like that. And then we can look at their action potential. So this is the action potential of the autorhythmic cells, where we don't see a resting potential really. It's always depolarizing and repolarizing, generating that rhythm. You need to know the different ions that cause the different parts of this curve, right? The slow depolarization, the fast depolarization, and the repolarization. But what Nikolai was asking about was kind of this. So when we look at our contractile cells, their action potential is a little bit odd. It is basically this. So their action potential is shaped like this, right? So we have, again, sodium as the main ion that's causing depolarization as it enters the cell. We have calcium that gives us that plateau entering into the cell. And then we have potassium exiting the cell, which causes the repolarization. But that plateau that we have there, number one, it gives us a protracted and extended contraction period, right? For skeletal muscle, it's okay for us to have little twitches in skeletal muscle, for it to go bzz, bzz, really quick and then relax. That's okay, but that's not okay in the heart, right? We don't want the atria going, and that's it. Or the ventricles are just going, like that, right? It's not going to give us an ejection of blood that we need. So we need a protracted contraction period so we can really squeeze the blood out. And then we need a protracted, basically extended relaxation period as well. We need that period called the refractory period where we can, those cells, even if they were stimulated again, they're not going to respond. They're like, no, I'm busy, let me relax, let us fill again, so that when we contract again, we have blood to be able to eject. So it has a longer action potential or contraction period because we want to fully eject the blood. And it has a protracted or elongated refractory period where it's not going to respond because we want those chambers to relax and fill again. Okay? So normal action potential is just like boop, boop, like that. It's like 5 milliseconds. That's it. This is like anywhere from 250 to 300 milliseconds. It's like a quarter of a second, right? So it's much, much longer. It's like 50 times as long as a regular action potential because we want a nice long squeeze and a nice long relaxation period as well for filling again. All right? Okay. What else? Yeah?
Can you go over like the angiotensin 1, like angiotensin 2, and like all that process?
Yeah, good question. I like that. So when we were finishing the hemodynamics section, when we were talking about how we regulate blood pressure and things like that, we had gotten into this idea that there are a variety of ways that we can tell our blood vessels when it's time to contract, when it's time to relax, and how we can really control that blood pressure. So I'm kind of looking along here. So long term, short term. It's a whole long section because we have figures in there as well. But this one at least lays it out for us in terms of the ways that we control blood pressure, right? So there's 3 levels of control, and this is the thing that we're going to see regularly with all of our systems. So we have our 2 kind of master and puppet systems of the body: nervous system, brain and spinal cord, and endocrine system, those endocrine glands that are releasing hormones, okay? So in terms of short-term regulation, how we can affect blood pressure, nervous control and hormonal control are really fast. We release these hormones like epinephrine, and now the heart's like really kicking over. Or we have long-term regulation as well as a third type of control, which is from the kidneys. And that's where we are filtering the blood and we're adding things to it, or removing things from it, but that takes a little bit longer. So anything we do to the blood definitely will affect blood volume and concentration, which affects blood pressure. But that control mechanism is a little bit longer. So you've got to be able to describe, kind of name and describe each of these. What is neural control? What's hormonal control? What's renal control? And then finally, there's like this localized control called autoregulation, where outside of what the main boss is telling you, you're like, I got my job, I know how to do my job, and if I need to speed up, if I need to slow down, I know how to do that. That happens as well when we have local chemicals that are released, like if you get a scrape or a bug bite or something like that, there's going to be changes in local flow to that area because of some of these local chemicals that are released that can also cause blood vessels to constrict or to dilate. Okay? So, in short, when we're talking about neural controls, that's when we're talking about the autonomic nervous system, where we have sympathetic and parasympathetic stimulation going to these blood vessels and to the heart, right? Sorry about the lack of penmanship there, but you get it, right? So sympathetic, if we're talking about the heart, sympathetic is fight or flight. It's going to speed it up. It's going to speed up your heart rate. It's going to speed up or basically strengthen your contraction as well. So you're going to get a harder squeeze during stroke volume versus kind of a weaker squeeze. In terms of the blood vessels, so you can do sympathetic and parasympathetic with the heart. Speed it up, slow it down. For blood vessels, interestingly enough, most of the blood vessels, you only have sympathetic control. So basically, when you stimulate a blood vessel, sympathetically, it constricts, which increases blood pressure. And then when you don't want that blood pressure increase anymore, you just stop stimulating it, and it naturally dilates and relaxes. Now the blood pressure goes down. So for that, it really only has a gas pedal, right? You step on the gas pedal, we constrict. With sympathetic, now when we're done, we just kind of let it relax again. So it doesn't have that sympathetic and parasympathetic in a lot of cases. Hormonal controls, you just have to have some examples of hormones that affect blood pressure or blood flow, right? Whether they have an effect on the heart or the blood vessel or whatever, have some examples of that you can share. So for instance, Orlando was talking about some chemicals and hormones that can affect that. So we see here some chemicals that increase. This is our increase of blood pressure. And then we have some examples afterwards of chemicals and notably hormones that decrease blood pressure. So norepinephrine and epinephrine, those are some of your easiest examples. That's like your adrenaline, right? Antidiuretic hormone, you should know that one too. So that one, remember we said that one's basically no pee hormone, right? So if your kidneys are holding on to fluid rather than letting it go, that's going to maintain blood volume, which is going to maintain blood pressure. If we don't have antidiuretic hormone, then your kidneys are like, we don't need this. Okay, we can let it go. That's going to reduce your blood volume, reduce your blood pressure. So antidiuretic hormone is going to cause not only just to hold fluids, but it also causes vaso blood vessel constriction. This is the one that Orlando was talking about, angiotensin II. There is what's called a RAS mechanism. It's renin that you see here, angiotensin, which you see here, and then lastly, this one is aldosterone system. So this is a hormonal control of blood vessels. It starts with renin, which comes from the kidneys, right? The kidneys know what's up with the blood. They're always looking at the blood and trying to figure out changes. Is the blood pressure too high? Is it too low? Do we have enough red blood cells? Do we need to make tweaks with that, etc.? In fact, erythropoietin that we talked about, which stimulates the red bone marrow to increase red blood cell production, comes from the kidneys. They're the ones that are looking at the blood all the time. So renin released from the kidney when we have basically too low a blood pressure. Renin gets released, it goes over and causes the activation of angiotensin, which is from the liver. Angiotensin then gets activated and activates what's called angiotensin II, which is from the lungs, of all places. And that is a vasoconstrictor. So angiotensin II, when that gets activated, it causes vasoconstriction, which also increases blood pressure. Okay? So angiotensin II itself has that ability to increase blood pressure, but it also causes the release of aldosterone. Aldosterone tells the kidneys to hold on to sodium. So, like if you're studying late or something like that, and you're eating salty potato chips to stay awake or something, or popcorn, what happens is you get kind of bloated afterwards. You're like, oh, that wasn't so good. Happens to me as well, like when I'm grading, my go-to is Cheez-Its. So I'm like eating Cheez-Its and grading and stuff like that, and it just kind of keeps me awake. It's not so good, right? But you got to do what you got to do, I know. So in any case, right, when you increase sodium levels, that creates an osmotic draw. So you load sodium in, and water tends to follow, right? So if we tell the kidneys with aldosterone to hold on to sodium, then it allows them to hold on to water too, which also works to increase blood pressure, okay? So that's also part of that, what's called a RAS mechanism, the renin-angiotensin-aldosterone system, which is a response that works to increase blood pressure, increase or maintain blood pressure. Okay? Good? Okay. So these are some hormones that you could use if you are talking about hormonal control of the cardiovascular system and blood pressure. You don't have to know them all, but you should know at least one or two hormones that have an effect on blood pressure and blood flow, or even cardiac output for that matter. So those are some that increase it. There's another one that decreases it. For instance, like ANP that we were talking about, etc. What else? So then it gets into longer term mechanisms, things like that. There was another section. Oh, I wanted to review this a little bit. So this one is talking about that auto-regulation where the activities that are going on in a specific tissue in a particular area of the body can also affect local blood flow and local blood pressure. So what this lists, all those little things that you see listed there, are different things that can either cause vasoconstriction or vasodilation at the tissue level. So there's another question in the exam objectives where it says, okay, talk about each of these different factors and lists a few, and tell me, what is that going to do to blood pressure or blood flow? So if that particular factor goes up or goes down, what's the response going to be? Are we going to get more blood flow to that area, or are we going to get less? And what I usually think of, it makes it easier for me personally, is I think of, okay, if I have a skeletal muscle that's working really hard, what's it going to be like in that local area? Well, it's going to see a drop in oxygen in that area because we're using oxygen to make ATP and stuff like that. So that's going to increase blood flow to that area, right? So if there's a drop in oxygen, blood flow is going to increase to that area. It's going to cause our vasodilation. If we see an increase in carbon dioxide, again, cellular machinery cranking out ATP, it's going to be releasing carbon dioxide. An increase in carbon dioxide also indicates that you have an active tissue there that we should increase blood flow there, right? A drop in pH, so say a muscle is producing lactic acid, etc., or more carbon dioxide, that's going to make it more acidic. That type of situation in that tissue is also going to cause vasodilation, more blood flow going there. Increase in heat. If a temperature goes up, that causes an increase in blood flow, right? So if you put a warm compress on something, that's going to bring blood to that site. If you put a cold compress on something, that causes vasoconstriction, right? So if you have too much inflammation and you hurt your ankle and it's swelling, you put ice on it, that constricts the blood vessels so it's not swelling quite so much, right? So temperature can have an effect on that as well. So look at that question for different local factors and how they can affect blood pressure and blood flow. And again, you don't have to like write it out in terms of like a paragraph or what heat does, but just basically say, when that factor goes up or down, what happens to blood pressure or blood flow? So if temperature goes up, we increase the blood flow to that area. If temperature goes down, we decrease the blood flow to that area. It's just as simple as that, of describing how each of those factors could affect blood flow. So you could say increase or decrease blood flow. You could talk about vasoconstriction or dilation. You could kind of describe it either way if you want. Okay? Other things.
Just to clarify, when you said, when like the question described the formation of different blood cells is initiated, is that referring to the hormone release?
Right.
The precursor is referring to the chart, like.
Exactly. Yeah. So when we're talking about those different blood cell types, red blood cells, white blood cells, platelets, etc., we want to know what's the chemical signal that tells the bone marrow to release more of that particular, or produce more of that particular cell. So erythropoietin for erythrocytes, our cytokines or interleukins for our leukocytes, and then thrombopoietin for our thrombocytes. Those are the chemicals that actually tell the bone marrow to produce those three different cell types, okay? And then, like you were talking about, kind of the family tree picture is, okay, if we need to produce it, we start out with that hemocytoblast, and then if we're going to be producing lymphocytes, we would then do the lymphoid stem cell, and that gives rise to our lymphocytes. For our other ones, we're going to go for the myeloid stem cell instead. So those are the precursor cells. And we don't have to know all of them. If you look at that slide where it has that big family tree, there are red boxes on any of the ones that you do need to know. You don't have to know all of them. Okay? So let's do this. Let's mix it up a bit because we've been doing a little bit of Q&A. Let's take a look at some example questions that also may just kind of get us in the mode of thinking about, you know, what types of questions we might see, topics that we might see, and stuff like that. So we don't have to do eye clickers. but we'll just kind of go along and think about these and we can kind of answer them together. Okay? Okay. So, let's look at this one. This one says, which of the following are cells found in the blood? So you think E in there? M. Okay, so that's pretty good. So we see erythrocytes, we see leukocytes, that's red blood cells, white blood cells. Remember that thrombocytes is the technical term, medical term, for platelets. Make sure that you know those technical terms as well, so that you can recognize what we're talking about there too. In general, sure, E is correct. Now, the only thing that I highlight with this is every once in a while, you may see a question where it says, okay, talk about the cellular elements or the cellular component of the blood, and it doesn't just straight up say cells. And that's because, remember that the platelets or thrombocytes are actually cell fragments. They're not whole cells, right? So that's why, don't get thrown off when it says cellular elements or cellular components of the blood, that includes our platelets. But if we're just talking about whole cells, well, technically the platelets aren't whole cells. So I'm not going to have like a super tricky question like that on here where it's like, oh, D is actually the correct answer because C, platelets are not whole cells, and that says cells. But it is a reminder in terms of, that's why they often refer to cellular elements as being those cellular components of the blood. Okay. Heart sounds. So we did talk about different ways that we can assess what's going on in the heart. We talked about listening to the heart. We'll be doing it in lab coming up. What do we think in terms of heart sounds? What do you think? Those are longer answers. I know it takes a while to read. So when we look at A, A is the ECG. It is a way of assessing what's going on in the heart, but electrical activity of the heart, right? So when we're listening to sounds, that's going to be listening to mechanical activity, movements, etc. So A is not going to be the answer right there. Okay, so it's a different type of test. B and C have to do with cardiac valves. So then you're looking at it, you're like, okay, so there's a few answers that have to do with that topic. So that, even if I don't know anything, it looks like maybe, right, that might be the topic that we're addressing at least. So we talk about valves. And so, so the answer for heart sounds does have to do with valves. But the question is, so we have our atrioventricular valves, and then we have our semilunar valves. So we have two sets of valves that are going to be opening and closing at different times. The blood flowing through them is what pushes them open, and also pushing back the other ways causes them to close. So the opening is a very smooth action, right? That just pushes, we saw those little membranes, it's really flexible. It's easy to push those out of the way. But when it pushes on the other side and it causes them to close, the blood gets really angry, and it's like doing this turbulent mixing right next to the valve, and that actually makes noise as the blood's going before it turns around and exits. So the closing of those valves and that turbulent flow of blood next to it as they're closing is what makes the noise. So not the opening, but the closing of those cardiac valves is what actually makes those loved up sounds that we're going to be listening to in lab. Okay? And the first set are the AV valves, and the second sound comes from the semilunar valves because the heart and the blood's passing through those different chambers through the cardiac cycle and through those valves in that way. Okay. The sites of exchange between the blood and the tissues are the what? Capillaries. Yeah. So this highlights two different things. Number one, it highlights the fact that we have different blood vessels that have different functions. Okay? So like we were saying, you got to know the structure of each of those different types of blood vessels and why they have that structure. Is it to resist all of that high pressure flow? Is it to help to move? Is it to help to protect the capillaries, like the arterioles have that smooth muscle layer that's relatively thick that protects the capillaries, etc. The capillaries are the thinnest and most numerous blood vessels. They are the site of exchange. Exchange of respiratory gases, of nutrients and waste products, but also fluid, right? Water is going to be exchanging there as well. So a key thing about that is that we spent a decent amount of time talking about those capillaries and how exchange occurs. So he said for a lot of substances like gases and nutrients, it's just diffusion. It goes from high to low, diffuses out. Waste products diffuse back in. But bulk flow, the movement of fluids, that was a bit more of a complicated topic. That's one of your bolder topics where it talks about hydrostatic pressure pushing it out, osmotic pressure at the end of the capillary sucking it back in, right? So make sure that you review that a bit in terms of capillaries and what's going on there in that exchange. Okay. Which part of the arterial wall contains smooth muscle? So the intima, that's a covering and lining, so that's going to be epithelium, right? So it's going to be epithelium, covering and lining, always. Media, think about the heart wall. You need to know the 3 layers of the heart wall as well. The middle of the heart wall is also muscle. So that's our myocardium in the heart, and here it's the tunica media. This is smooth muscle, but the middle is going to be our muscular layer. The outer layer is mainly going to be connective tissue. It might be elastic fibers in there and stuff like that. It's basically kind of holding it together. So our tunica media there is going to be where our smooth muscle is. Now the amount of smooth muscle is going to change, the thickness of it, depending on whether it's an artery or a vein or something like that. But for those larger vessels, any vessel that has smooth muscle is going to be in the middle of that wall. So this is just reminding us of being able to compare the structure and the function of our different blood vessels, kind of why they're structured that way. Oh, how about this? Back to blood. We did blood typing. So Jason has circulating anti-B antibodies, but not anti-A antibodies. Which blood type does he have? Now, this is not considering the whole positive or negative or anything like that, just the letters themselves.
A.
So that would be type A, okay? So if we remember, when we're looking at it, the antibodies are part of your body's defenses, right? We'll learn about that with the immune system as well. Antibodies are there to attack foreign cells. So if you have antibodies against the B antigen, your cells better not have that B antigen, or your own antibodies are going to stick to your own cells, and you're going to have an autoimmune disease. You want antibodies against foreign cells. So whatever you have antibodies for, you're not going to have cells that have that. So in the upper left-hand corner, we can see somebody with type A blood with A antigens on the surface of the cell, and then anti-B antibodies floating around, okay? Somebody who has type O blood has both of the anti-antibodies because their cells have neither A nor B antigens, and type AB blood, since they have both antigens, don't have either antibody floating around, because if they did, it would stick to their own cells. Okay? So for our different blood types, you've got to be able to describe what's an antigen, what's an antibody. The antigen's always stuck to the cell. The antibody's floating around. It's that snowflake-looking thing, that protein. Okay? Like our immunoglobulins, antibodies are floating around, plasma proteins. And you need to know which blood type has which type of antigen and antibody. Okay? So make sure you practice a little bit with that as well. ECG, there we go. So QRS complex of the ECG is associated with E. So this one, when we look at our ECG, remember that the smaller waves, there's only one thing going on. So the P wave is atrial depolarization. The T wave is ventricular repolarization before they're relaxing and filling. But the QRS wave is extra big because there's 2 things going on there. The atria are repolarizing, and the ventricles are now depolarizing at the same time. So that makes that wave that much bigger. So when you're describing your ECG output and those different waves, if you're talking about your QRS wave, make sure that you talk about both things occurring there. Atrial repolarization as well as ventricular depolarization. How about this one? The differential count of a person suffering from both allergies and parasitic worms would probably reveal elevated numbers of. So this is helping us to review. We have 5 different types of white blood cells, each one with a different function to help us protect against different types of pathogens. So we're basically addressing 2 different ones. What do you think? So even if you didn't know, so you're going to want to, make flashcards or something like that for each of your different white blood cells and then what they do on the back. But even if you didn't know that, to a certain extent, right, this says we're addressing 2 different attacks on the body. So probably we're going to have 2 different white blood cells. And so when you look at those answers, technically D is the only one that has 2 white blood cells included into it, right? So, you couldn't totally count on that. It's better to know what the function is for each of the blood cells, but that's a way to kind of look at your answers and kind of reason things out as well. So, eosinophils are associated with parasitic worms. That's one of their main functions. So you're going to see a bump up in eosinophils if you have a parasitic worm. Allergies, the basophils release histamine. Remember, we take antihistamines when we're having an allergic attack and stuff like that? Histamine causes vasodilation, causes inflammation. So if you're having an allergy attack, you want to take some antihistamines that's going to decrease the activity of those basophils. It's going to kind of get rid of that histamine which is causing that. So it would be our A and B as correct there. So that's making sure that you can align the function of those cells with the actual cell themselves. Yeah.
It's kind of unrelated, but would an EpiPen have potent antihistamines in it, or is it just the epinephrine?
So it's just the epinephrine. So the strong effect of that hormone, as we were talking about before, not only affects the heart, right, in terms of heart rate as well as strength of contraction, but it also affects the blood vessels. So it causes them to vasoconstrict. So it's more of like a mechanical response in terms of jacking up blood pressure when it's dropped really critically low, as opposed to, okay, things are leaking out, let's kind of shore them up and not quite leak so much. We're going to just kind of increase a lot of the mechanical activity instead. Yeah. Okay. All right, along with those blood cells like we were talking about, this is that whole kind of cell line that I was mentioning where we've got our different kind of cell lines. The only additional ones that we need to know, there's our hemocytoblast, myeloid stem cell, lymphoid stem cell. Megakaryoblast you need to know for your, you can probably just do megakaryocyte, is your precursor to your platelets. And then the reticulocyte that we see here is our direct precursor to our erythrocytes. All these other ones, you can just say basically myeloid stem cells give rise to these. And lymphoid stem cells gives rise to our lymphocytes. Right now, we're not really worrying about T and B lymphocytes. We'll get into that as we get into our immune system. Okay. Ooh, how about this one? Ooh, it's a lot of words. So which of the following is correct? NFP is basically net filtration pressure. Like when we add the forces together, do we have a big movement that is out or in altogether? So that's net filtration pressure. So we're talking about fluid exchange at the capillaries. Which one?
I think it's E.
So E, basically kind of like an all of the above type of thing? So that's a good question. Any other guesses? We agree with that? Well, let's take a look. What's that?
I say B.
Okay, so that's one part of it, yeah. So this is a figure showing the same thing that we were talking about before in terms of the exchange at the capillaries, right? This one actually shows some of those lymphatic capillaries in there as well. That green thing is not alien. We actually have lymphatic capillaries next to all of our regular cardiovascular capillaries that are there to pick up some of that excess fluid that we had talked about that gets left behind. So to be sure, C, where it said lymphatic capillaries pick up excess fluid, that's true, right? Because there's an uneven kind of distribution of fluid. We have a little bit more fluid going out than coming back. But here's the key thing, is that at the beginning of the capillary, we have a net movement out, so that we can be delivering fluids because our hydrostatic pressure is higher than our osmotic pressure. So overall, we have a net filtration pressure that goes out at the beginning of the capillary, and then by the end, our hydrostatic pressure has weakened, and so now that osmotic pressure is more than our hydrostatic pressure. So overall, when we add those together, we have a net movement that's actually coming back in by the end of the capillary. So what we see at the beginning of the capillary, we have delivery to the tissues, and by the end of the capillary, we have fluid coming back in. And is that what that says? This one says, at the beginning, at the end of the capillary, it says in. That's wrong. At the venule end, so this is at the beginning. At the beginning, it says it's coming in. It's actually going out. And this is the end, and it says it's going out. So what we're seeing there is basically a reverse of what it should be, right? So by the beginning of the capillary, we should have fluids being delivered, and by the end of the capillary, we should have fluids coming back. So really, out of all those, basically just C was correct. Now again, that was a lot of words. I wouldn't have the question be that complicated for a multiple choice question, but the topic itself, in terms of fluid exchange, is a bigger topic that you do have to be able to explain. Like we said, the role of hydrostatic pressure or blood pressure, the role of that plasma osmotic pressure, drawing things back in, and how we have movement out of the capillary at the beginning because the blood pressure is higher, and how we have movement back in to the capillary by the end because now the osmotic pressure between the two is higher instead. Okay? So, definitely take some time with that one. Ooh, how about this one? Blood passing through the bicuspid valve will enter the. So you got to think about where is that bicuspid valve and where is it separating. So if it passes through, where is it going into? Yeah. So tricuspid valve, it's right to try things. You should try new things, stuff like that. It's the right things to do. So tricuspid on the right to try. Bicuspid on the left. So bicuspid's on the left. If we pass through that one, we're going into the left ventricle. Okay? Okay. So make sure you know your basic anatomy too. Even though we have a lot of physiology, a lot more physiology on a lecture exam, we got to have that basic understanding of the anatomy as well. So you will have some anatomy questions on our lecture exam as well. Okay. So that was just kind of a way of reminding us of some of those major topics there. So in the meantime, we have our lecture quiz tonight. So make sure you get that done. Again, Respondus Lockdown Browser, but you can have other things available to you. Totally fine for you to have other electronic resources or printouts available. Saturday by midnight is when you have your exam due. Closed book, closed note. Make sure you have a good internet connection, that people in the house know you're taking a test and not to bug you, that it's going to be nice and quiet so you can focus. If something happens, right, do everything you can to prepare to make a smooth exam testing session for you, right? Let people know not to disturb you, have everything set up. If you want a drink of water or a snack, that's totally fine to have there, right? Get it, everything set up. But if something happens, unforeseen, right? Your dog gets up, who's next to you, and throws up in your lap or something like that, right? If something like that happens, take care of it, and then just basically tell the camera, oh my gosh, sorry, I got to duck down here, and I got to wipe this up for a second, and then I'll be right back, okay? So just tell the camera, because Respondus is going to flag it. It's going to send a flag to me saying there was something weird. You disappeared out of the screen or something like that. It's going to tell me that. And so that way you can just let me know what happened so that I can be assured that everything's fine. If it's something big, right, and you actually have to stop the test, then, shoot me that email afterwards and we can discuss, what we can do to kind of get around that. Okay? All right. So early lab people, you are free to go. Late lab people, I will see you in a short bit.